Good morning, students. Let's continue with the chapter immunity. Well, today going to study the next important topic, which is antibodies. As we are now familiar what are antigens, then the antigen binds with the antibody. So the antibodies are immunoglobulins which are produced in the body in response to antigen. So these antibodies may found bounded to a cell membrane or remains free. Antibodies are produced by B lymphocytes or B cells and plasma cell. We have already discussed. Next, antibodies direct the antibody mediated immunity, also known as humoral immunity. Next is the structure of this structure is Y-shaped. This comprises of two chains. This is heavy chain covered by two half. These are called as light chain. They are joined by disulfide bond. Heavy chain, and in turn heavy chain and the light chain is in turn bounded by disulfide bonds in both the sides. The one terminal is known as N. This is known as N terminal, while this is the C terminal. N terminal and this. These are the antigen binding sites. Antigen binding sites. This is the light chain and this is the heavy chain. Here it is a variable region. From here to the last is the constant region. This antigen binding site is known as FC region. This is the structure of, and these are the disulfide linkage. This region is known as hinge region, which are responsible for flexibility of immunoglobulin. So these are the different parts which is found in the antibodies. Now, second, we are going to study the types of antibodies with their functions. First, we are going to start with the 5 types of antibodies: IgA, IgE, IgG, IgM, and IgD. Before reading these types of antibody, we'll study about the antibody structure which we have seen in the diagram. Antibody structure. With the antibody structure, the first part we're going to study is heavy and light chain. In the heavy and light chain, they are made up of 4 peptide chain. Out of 4 peptide chain, 2 chains are 2 small chains are called light chain, while 2 longer chain are called heavy chain. So we can represent this by light chain by L, heavy chain by H. So 2 light chain and 2 heavy chain, H2 and L. Next part is constant and variable region. Constant and variable region. There are 2 different regions, constant and variable, in the antibodies. In each chain. In each chain of the antibodies, as I mentioned in the diagram, there are 2 regions, constant region, variable region. The next part is disulfide bond. Disulfide bond and hinge region. A disulfide bond joins light chain with heavy chain. 2 disulfide bond link to heavy chain. The flexibility of antibody structure is due to hinge region. Due to this only, the structure of antibody is Y-shaped structure, and it is mainly made up of proline amino acid, which cling or bend in the structure. Next part is Fab, which is a fragment antigen binding, and FC, which is a fragment crystallizable. So the Fab, fragment antigen binding, and FC regions. First, the two identical fragments of Y-shaped sites, thus named fragment binding antigen binding of Fab region. Next, this Fab region, antigen bindings region, bind to specific antigen in key and lock pattern, forming antigen antibody complex. The next region, which is called as FC, lacks flexibility and ability to bind to antigen and can be crystallized, therefore called fragment crystallized or crystallizable, more appropriate, the FC region. This is all about the antibodies. We're going to detail and mean the structure of antibody. In the next chapter, we're going to study about the type of antibodies in detail. Thank you.